So welcome to Virtually Marine Mammals, brought to you by the Marine Mammal Foundation. I'm Dr. Kate and I'm the Director of the Marine Mammal Foundation. So today is our very first session of 4 that we're going to be running, and it's Dolphins at your desktop. So we're going to be taking you on a bit of a journey as to how I got to where I am today and the discovery of a dolphin and the amazing things that the Marine Mammal are doing, and the Marine Mammal Foundation is doing. So I'll be sharing a PowerPoint presentation with you, and we'll go from there. So I hope you enjoy it, and we'll get to the Q&A session at the end. Welcome to Virtually Marine Mammals, Dolphins at your desktop. As I said, I'm Dr. Kate, and so the Marine Mammal Foundation is a really fantastic organisation that was established in 2013, and its vision is to be one of the leading conservation organisations protecting marine mammals in their environment through our research, community engagement and education. So the foundation was set up so that we weren't just a research organisation, that we were using the research that we're doing to be able to create greater awareness, to be able to have stakeholder input, but also to help guide policy and mitigation management for some of the threatening processes that we have out in our marine environment. So we have an amazing team of people. So I'm the director of the Marine Mammal Foundation. We have a great set of researchers that on Thursday's session you'll be able to meet. So we have Chantel, Rachel and Marinda. As our researcher, Hayley's popped herself up in there too. And we also have an amazing team of educators. So we have Matt, Caitlin, Amber, Hayley, Bridget and Jacinta. So we have an amazing core team of people there. You'll probably notice that the majority of those are females. So Matt's very lucky to be working with us all. So not only that, that's our core team. We do have an advisory board with some amazing people that help advise the foundation. And we also have a really great set of volunteers that help us do what we do, from outreach and education to being on the boat when it's freezing, to citizen science, community festivals, our Citizen Science Burrunan Watch program, our Young Marine Champions. So we've got a really great set of people. So a little bit about me to start off with. I did my Bachelor of Science degree at Monash University. And I started my honours project in 2003. And then from the results of the project, I was able to obtain the Dean's Postgraduate Research Scholarship to continue on to my PhD, of which I did and completed in 2012. I have over 16 years experience with marine mammalogy. I'm vet trained to conduct gross necropsies and scientific sampling on dolphins and whales that wash ashore, and I've conducted a number of those across Victoria and Tasmania. I've been a lecturer at Monash University, research associate with Museums Victoria, Monash and Deakin. I was the former president of Australian Marine Sciences Association in Victoria, so it was the vice for 2 years and then the president for 2 years, and handed that off to Cade now. And my current role is the founding director of the Marine Mammal Foundation. So quite a lot of experience out there on the water, but just to put it in context. So my journey, I've always said in my conservation biology lectures, that change starts from one small action. So every waterfall has to begin with that one drop. So my one drop for me was when I started to dive out there in beautiful Port Phillip Bay. This is obviously not Port Phillip Bay, but diving evokes such a huge passion for me and in the marine environment. I found that place where I fit, and I was at peace, and it was an amazing experience, which I'm sure you've all experienced, those sorts of feelings. And so from there, I really didn't want to just go for a dive and say, that's a pretty fish and great coral reef and great sponge garden. I wanted to learn more about the marine environment and how things interacted, and more importantly, how I could make a difference to protect that beautiful environment. The next thing that happened, I was introduced to the amazing dolphins in Port Phillip Bay and the Gippsland Lakes. And I've yet to meet a person who doesn't like a dolphin, so I'm not alone there. You know, when these dolphins choose to interact and bow ride and you make that eye-to-eye contact, you know, sometimes you're wondering who researches who. But that was the next step that really evoked a passion and conservation concern. Now, during my undergraduate degree, I also got a lecture in the use of DNA for cryptic species, which, of course, dolphins are, as they spend most of the time under the water. So the DNA was the really big key that I could use as a tool to understand these dolphins that were in Port Phillip Bay. So I'll let you just watch this beautiful video. This is using one of our newer technologies of a GoPro camera. And these are the beautiful dolphins of Port Phillip Bay with their inquisitive nature. You can hear those sounds there. And off they go. And that kind of interaction is amazing. They came over and checked us out and then headed off. So obviously that instilled a massive passion in me. Now, bottlenose dolphins in general are under the Tursiops species lineage, and there's been upwards of 20 Tursiops species described, and these vary in a number of different qualitative and quantitative traits. And they're in this evolutionary complex set up with a number of these different dolphins. So it's quite an interesting study species to have a look at. Now, Tursiops show different genetic and morphological differences. They have different morphotypes, but they're not consistent across the different regions across the globe. Historically, in Australia, all bottlenose dolphins were classified as Tursiops truncatus, the common bottlenose dolphin. But this has been blurred by the use of genetic analysis. In southern Australia, here where we're based, we have a distinct inshore and an offshore type. So the inshore type is around about 2.5 meters. You can see it pictured here, and it's in these semi-enclosed embayments or estuarine areas. And we also have an offshore type, which is about 3 meters and a really large, dark, robust animal that's found out in Bass Strait. So here we go, the common bottlenose dolphin, Tursiops truncatus, is really large in our area. Big tank, dark animals are pretty robust animals. And we have the Indo-Pacific bottlenose dolphin, Tursiops oduncus. Now, my question when I first started to have a look at these animals, what is this? This is one of our dolphins in Port Phillip, in Gippsland Lakes. Now, it's a bit of a mishmash between those two different species. So you can see that it has a stubby rostrum and the falcate dorsal fin like the common bottlenose dolphin, but also had the gradation of grey like the Indo-Pacific bottlenose dolphin, but then also has different characteristics that weren't really common to either of those. So that was my first question when I first started, was which dolphin species? And at the end of the day, to cut the long story short, which I'll go into in a little bit, they turned out to be neither of those. And in fact, using multiple lines of evidence, we were able to publish a new dolphin species paper back in 2011 and formally described the Gippsland Lakes and Port Phillip Bay dolphins as a new and separate species called the Burrunan dolphin, with the Latin name of Tursiops australis. Now, Burrunan comes from the indigenous narrative from 4 of the main dialects across the Kulin Nation. So it was fitting as a historical reference to these dolphins in the area. Now, the DNA was really the key. So we can extract different sequences across the DNA and then go on to this beautiful geek's paradise of GenBank and download that same sequence from multiple different species across the entire globe. And then you can build a phylogenetic tree. So without having to understand the whole DNA process behind that, you can see that there are a number of different dolphin species that not only look different, but have different DNA. So I was able to extract the DNA from these particular regions and build them into a worldwide phylogenetic tree. Now here we see the common bottlenose dolphin and the Indo-Pacific bottlenose dolphin. So I had the hypothesis that it would fall out with either of those two species. But in fact, the first round of DNA, it fell out actually quite differently. It wasn't even within those Tursiops lineage. In fact, it had a number of other different sister taxa that looked nothing like Tursiops species. You can see it all the way up there, separated out of that Tursiops lineage. Now, I also had a look at the cranial morphology. So I went to numerous different museums across Australia and got my hands on as many bottlenose dolphin species as I could, and took standard cranial measurements and tooth counts, as well as qualitative traits. You can see the two skulls there, A represented by Tursiops truncatus, the common bottlenose, and B represented by Tursiops australis, which we now know as the Burrunan. I also had a look at the external morphology, so what they look like on the outside, and completed a number of different measures from deceased dolphins. I was also able to collect the genetic samples of those to match where they were falling out genetically to where they were falling out morphologically. A whole bunch of stats later, and we could see that they were separated. In the green there is Tursiops oduncus, in the blue is Tursiops truncatus, and the red is Tursiops australis. So we can see it separating out on both of those areas. Now, at that stage, that was 3 lines of evidence, all leading towards the fact that these were different animals. We also had a look at diet, and we've continued our studies. At that time, we were able to show that multiple lines of evidence represented a brand new dolphin species, and again called Burrunan dolphin, Tursiops australis. So that was my journey of my PhD. Since then, we've had these newer technologies that have been able to, rather than look at very small fragments across the DNA, which is what I used during my PhD, to have a look at entire mitogenomes. And this is a paper that was published having a look at the mitogenome. And we can see our common bottlenose dolphin, our Indo-Pacific bottlenose dolphin, and again, separated by numerous other sister taxa is our Burrunan dolphin. So again, looking at these very large regions, we were able to see that these animals were in fact very different again. Now, one of the interesting things with this is this study was able to have a look at when that split was. And that split within that Tursiops lineage was defined at over 1 million years ago. And the deepest node for Tursiops has that strong support for the Australasian origin, which you can see here, the Burrunan splitting off to the other Tursiops species. Since then, we've had a number of different studies, and I've also had a look at, I've now got a huge amount of full genomic information, and it is all, again, pointing to a different species. So that species status is recognised across the globe, and a number of other newer studies have come out verifying and validating the Burrunan dolphin, which is super exciting. You know, the fact that in 2011 we didn't know that they were separate, to 2013, you know, being recognised as something special and listed as endangered, to now knowing that they're the most ancestral node is pretty cool. Now, obviously, that started that one drop of being under the water and loving, you know, that marine environment, started this passion and this flow and this knowledge, and my waterfall was sort of well and truly flowing by that stage, which is great. But my next step was to keep going with my waterfall and establish the Marine Mammal Foundation, which is something I'm super proud of. Now, this allows us to still conduct those amazing applied research initiatives that you generally see within a university setup, but also allows us to be able to get out into the environment and do things like this and talk to people about how amazing that environment is, with the desire to be able to protect it and conserve it for future generations. So at the Foundation, we have a number of different research programs operating from population modelling, social alliances, all the way through to having a look at the threatening processes of things like impacts of vessels and genotoxicity and an ever-increasing noisy soundscape. Now, we do that based at the Foundation, but we also have a number of amazing partner organisations and universities and government organisations that allow us to do this work. So a little bit about our flagship project, which is obviously related to the Burrunan dolphins, Project Burrunan. So what do we know about the Burrunan? We know it's described as a separate species. So our common bottlenose dolphin pictured here, you can see the distribution is all across the globe, tropical, temperate, inshore, offshore. Whereas our common Indo-Pacific bottlenose dolphins are restricted to the warmer waters of the Indo-Pacific region. Now, our Burrunan are restricted to just this very small part of Australia here, with genetic confirmation of the species being in Tasmania, Victoria, and South Australia. Here in Victoria, we have two populations, one in Port Phillip Bay and one in the Gippsland Lakes, and they're both incredibly small, with populations of only 120 and 65 resident dolphins. Looking at the DNA again, and without having to understand this fully, you can see the pie charts, and these are essentially the surnames of the dolphins. So the Smiths and Joneses live in Port Phillip Bay, or in the blues and purples. And the Gippsland Lakes are made up of a completely different genetic stock, which is quite a surprise because dolphins can swim. They don't need to. There's no barrier between Wilson's Prom and Tassie, but there's clear differentiation with the population. Now, what was interesting is that every haplotype or surname of these dolphins we found down in Tasmania matched those in Gippsland Lakes, but then very little gene flow the other way. In fact, 0.04 of a migrant per generation between those two populations. So we've been able to have, based on the small population sizes and the significant threats with living in heavily urbanised agricultural and industrial areas, the dolphins have been recognised as endangered under Victoria's FFG Act, and we're working hard to get them recognised under the EPVC Act, the Federal Act, and also the IUCN Red List. So we go out on our boat, there's Kirby, our research boat, and find our dolphins. And we can tell the difference between our dolphins based on these nicks and notches. You'll see a number of different nicks and notches along these dorsal fins. So we're able to identify each of those dolphins based on that. So this is Jimmy. So Jimmy is one of our Gippsland Lakes dolphins, so Jimmy Fornick. And you can see there's 4 distinct nicks in there. Now Jimmy's been a part of our resident population Gippsland Lakes since I've been studying them since 2006. And one day Jimmy was in the pod and the next day Jimmy was not in the pod. And then when we found Jimmy again, Jimmy actually had a calf. So Jimmy was in fact a Jemima. So we're able to tell the sexes of the females based on these associations. Now, Finn, who's Jimmy's calf, was born in 2013, and we've been able to track Finn based on this lesion that he was born with, the same way as I've got a birthmark on my face, and it's pretty distinguished from my characteristic. And we thought we were able to then track Finn through, and then through this leaping shot find out that Finn, in fact, was a male. We also have a couple of other really distinct individuals. This is Modi with her lump on there, which seems to be a deformity. And we have Tiger with her beautiful white stripes. So we've been able to track Tiger and Finn, who are our 2013 generation calves, all the way through. So we're now on 2nd generation calves of our research. We also have a look at Burrunan dolphin behaviour, so what they're doing when we're doing our focal follows. And this is just a short video on some of the footage we get from the Gippsland Lakes. Oops. You can see you've got this surging behaviour over onto the boat, and the water can be a little less than clear, but you can still get that beautiful interaction. You can see Chantal there with the GoPro in, which allows us to have a look at that underwater behaviour, but also to collect fin IDs and then also gender. Sometimes they're a bit tricky. You can see me up the front trying to actually get those dorsal fins. But that's a beautiful interaction when they make that choice. So during these focal follows, we look at their behaviour and we can see how they're using the different habitats under different times and on different group compositions, which is what we call focal follows. And we can also use that with geospatial mapping to have a look over certain feeding areas to see if there's correlation between these sites we're seeing them and their behaviour and then say seagrass density. We're also having a look at vessel and boat impact. There are regulations in place to protect the dolphins in that vessels, both powered and unpowered, are not to approach within 100 metres and jet skis within 300 metres. So obviously we can see a number of breaches to those, and you can see Lucky in the middle of that photo on one of our calves with those prop injuries. So we're having a look at that impact and the compliance of vessels to those regulations. We also have a look at mixed species association. So it's not just all about the Burrunan dolphins. We have other species that we research, the common dolphins in Port Phillip Bay and the Australian fur seals. So this is a bit of a fun interaction when we're having mixed species associations. So the other thing we're having a look at is the marine mammals of Victoria. So we've been able to, or Chantal, as a part of her honours project, was having a look at the cetacean biodiversity across there. So we're having a look at where we're seeing all of our marine mammals, not just our Burrunan dolphins and not just in these localised areas. We're having a look at our dolphin acoustics, and we're also having a look at marine mammal health. So having a look at how toxicology and toxicants within our system are potentially impacting on our marine mammals. So if you want to tune in on Thursday, you'll be able to meet the researchers and they'll go into more detail about these two amazing projects. So our research is obviously wanting to be with conservation outcomes. So as I said, we've had the dolphins listed as endangered, but we're also being heavily involved with having the assessment of the values of the ecosystem, and then also having significant input into marine mammal regulations. We've introduced our marine litter project. Obviously marine litter is a big deal and we spend a lot of time on the water, but a lot of that time is actually picking up plastic bags and chip packets and balloons. So, raising awareness about the impact we're having on the environment. We've got an amazing education and outreach program in which we go out to schools and now taking that online and out to festivals and really getting the next generation interested in conservation and in marine research. On that, we have our beautiful marine champions. So these are from grade 5 through to VCE, and it's a dedicated program to really foster their love and their connection of marine environment and in sustainability and conservation. We have our citizen science program in the Gippsland Lakes area called our Lakes Champions, and they get involved in a number of different activities from clean-up events, education events at festivals, but also are really instrumental in our citizen science program, Burrunan Watch. So how you can get involved? You can adopt your very own Burrunan dolphin or your seal. When you're out on the water, you can log a sighting via our track 'em function. Of course, remembering to keep those distances away and allow the animals to have the choice to interact with you. We are a very small not-for-profit organisation, so any donations or sponsorship or support goes a very long way. And with that, I'll just show you this pretty cool video in the Gippsland Lake so you can see the different system in Port Phillip Bay with the clarity of the water to having a friendly jellyfish as your neighbour in the Gippsland Lakes area. And important to note that all of our footage and our research is conducted under permit and strict animal ethics approvals. So together we can ensure what we do today creates that better tomorrow. And with that, I'd like to really thank you for your time and also thank our funding providers for allowing this to happen. Great. Well, thank you all for attending. We very much look forward to seeing you all on Thursday's session. Again, we'll send out that link so you can join in. Yeah, we look forward to seeing you then. Cheers for now.